So, hi everyone. Thank you so much for being here today. My name is Lila. I am a science manager at Animal Free Research UK, and today I'm going to talk to you about new approach methodologies and their relevance to human research and drug development. So first, I would like to start with an observation. I think we all agreed on that, but I wanted to just give you some figures that are quite striking to me. So if you look at the likelihood of a drug to be approved in clinical trials, so after having been tested on animals, it's lower than 8%. And if you look at the cost of a drug to be approved and brought to the market, it's about 2.6 billion American dollars. And it takes approximately 10 to 15 years for a drug to accept and be in use for health benefits. So I think it's clear that there is a real issue with the business of drug development, which high rate of failure. And I don't know if you would agree, but I think you would, that it's not a sector that you would like to invest in if you want to make money out of it. It's really a failure in cost and time as well. So during this presentation, what I would like to talk to you about is first trying to explain why we think animal studies are failing the research and drug developments, and then also to provide you some key information about what we call new approach methodologies, and also into a bit more details in the in vitro approach methodologies and in silico. And to finish, we will be discussing the current and future challenges and opportunities with these approaches that are human focused. So first, as I mentioned already, we think that there is a real issue, the business of drug development. If you look at the amount of human diseases that exist currently, it's about 1,000. We only have 500 of treatments available for these diseases, which means that there's still a lot of efforts to be put into research to develop new drugs. And in parallel to that, there is also, as I mentioned, a high cost, a high failure in terms of ethical and financial, because we waste a lot of lives. With at Animal Free Research UK, but also for a lot of other biomedical stakeholders, the reason this high failure is the too high reliance on animals. And if you look at the drug development pipeline, animals are actually being used in both the basic research and also the preclinical studies, so to test the efficacy and the toxicity of drugs being tested in human, in what we call the clinical trials. The issue with animals is, first of all, they have a very low productivity, as you could see with the failure of the high rate failure that I have mentioned. They also have a very low specificity because of the fact that they are not human. There is species in between. There is difference in between species, and so the specificity of animals is very low. And also there is an issue with reproducibility as well in animals. And I will enter into a bit more detail in the next slide. Something very important as well to consider when you just look at animal experiments like not human, and miss as well targets, meaning that something that doesn't work in animals can actually work in you, but you would just miss it because you've been testing on animals. And last but not obviously, considering all the above mentioned issues with animal experiments, it's not really ethically acceptable to use animals in the drug development process, knowing that there is such a high amount of issues related to the animal testing. And there's more and more evidence and figures that really show that there is an issue using animals, and it's not a good science to use animal. And I just highlight a couple of figures here. One really striking to me is like the fact that a 1/3 only of the highly cited research using animal are useful for human trials. So meaning that 2/3 of these studies are actually useless and do not benefit any, do not lead to any benefit to humans. Another important figure is the fact that animal studies overestimate by 30% the effectiveness of a treatment. So it's really showing that there is it's like animal studies and science related to animals are overestimating the quality of the and the translation power of the model. There's also something I mentioned earlier, which is the species differences. Even though, for example, between human and mice, the genome itself, the sequences of genes are 85% similar. When you look at the regulation of these genes, there is actually between 41 and 85% of differences, which definitely results in differences in processes and in disease mechanism. Additionally to that, some experiments show that there is also significant metabolic differences between human and mice that leads to obviously again differences in mechanism and differences in interpretation of the data. So all of this is really important to consider and really show that animal research also lead to misinterpretation of data and results. And it's a real issue when you then consider the drug development process. Additional evidence of the poor science are listed here. It's not an exhaustive list, but I wanted also to point out the fact that there is no best practice and a standard to study diseases in animal, on the opposite to, for example, human trials. There's also like an ignorance of the bias due to the lab environments. When you study disease in animal, you also do not consider the fact that the environment of the animal is also leading to stress, and the diet of the animal can also bias the result of what you are looking at. And a lot of studies do not consider the sex or the gender of the animal, which is a huge issue as well. And something that is more and more obvious and has been pointed out in a lot of publication is the fact that if you don't publish negative results, especially with animal studies, this is a real issue, because you are just wasting a lot of time and obviously lives as well, and all of this focusing on the wrong organism, because you're studying an animal that is not a human. According to the World Health Organization, in 2018, the two leading causes of death were cardiovascular diseases and cancer. And if you look at the likelihood of a drug to be approved for these two diseases, it's about 5%. So this really, to me, highlights the fact that there is a real issue and a real emergency of changing the way we are developing drugs to ensure that we improve this probability of success to treat actually the diseases. I wanted to show you this really important example of a lab that came to us saying that they actually look back at the work that they were doing. They are working on rheumatoid arthritis, and they looked at the publication of 10 years, and what they were really shocked thing is that about when we looked at about 10,000 papers publication on these diseases, represents almost 1 million of months, taking into account all the studies. It hasn't led to any drug approval. So this means that all time and money and animal use has been for nothing, and they actually realize that it is time now to try to develop new methodologies and new model to actually study better the disease, and models that are more focused on the human and less on the animal. And it is not an isolated example. There's more and more literature showing that there is a real issue with animal experiments, that they are not reliable enough and they are not relevant enough for the human diseases studies and the drug developments. So all this, I hope it convinced you that we need to change, we need to really focus on the human biology itself. And if you want to accelerate the breakthrough of drug development, we need to embrace models and to develop these new models that are focusing on human. So the issue with what we call the in vitro, so the study of a disease in dishes and in the lab, so not in the living organism, is obviously the fact that it's not representing the whole physiopathology of a disease or of the human body, and it is hard to replicate what's happening in the human body. But more and more models have been developed in the past years and are getting closer to what we would see in a human. So I would like to focus first on what we call the human pluripotent stem cells. That has been a great breakthrough these last years. So the what we call the human pluripotent stem cells are cells that are able to differentiate in any cells of the human body. So it can differentiate it in a heart cell, in a brain cell, etc. And what has been revolutionizing this field is the fact that we are not able to take any patient cell from the skin or for the blood and to differentiate these cells, so to make them what we call pluripotent, so to make them back to a cell that is not differentiated. And then with the specific protocol, make them differentiated in whatever cell we would need. And this is really a revolution for the study in vitro, because as you take a sample from the patient, you just recapitulate the genetic background of this patient using the induced pluripotent stem cells. So this technology has a lot of advantages. It's quite easy to generate. It's quite quick as well. It has low cost and it's really reproducible. And so because it's quite fast to develop, it's also a good technology for the screening of a drug. But obviously, as it's called a 2D culture, cells are not really in the same shape in the same morphology as it is in the human body. This is mostly layers, and you can add different cell types, but it's really limited. So you don't really recapitulate what we call the microenvironment of the body, and you don't have the same heterogeneity that you could in humans. So because of these limitations, you cannot really recapitulate a full organ disease if you use the in vitro induced pluripotent stem cell. But over years, and to like overcome the issue of the morphology and heterogeneity that you have in 2D, researchers have been developing what we call 3D models of stem cells or organoids. It's basically what we like any organ that is growing in vitro, but in 3D. So with a different cell type and also the closer morphology of what the cell has in the organs. And you can use also scaffold to recapitulate the matrix or the microenvironment of the in vivo organ. And as you can see in this pipeline over years, researchers have been really successful developing organoids for all different organs of the human body, from the gut to the brain to the placenta and so on and so forth. There is different approaches and different way of producing what we call the organoids. You can use cells from what, as I mentioned, the pluripotent stem cells or the embryonic stem cells, so cells that are not yet differentiated, and to differentiate them in using the different signaling molecules and factors to make them the cells that you will want. So it's using the induced pluripotent stem cells is very versatile because you can basically use these cells to grow any kind of organs that you would like. If you compare that to an organoid that is developed from the adult stem cell, so basically you just take a sample from a gut, for example, in the cancer research, when you resect the tumor, you can get a sample and make it grow in a dish, and this is what we call the adult stem cell organoid tissue. The two approaches are really complementary because they are different and they have their strengths and weaknesses. And depending on the organs and on the disease that you want to study, both are really can be really useful. And for example, for some organs like the brain, it's quite hard to get the adult stem cell tissue. So you basically can grow brain organoids only from the pluripotent stem cells. And there's a lot of reason why organoids are being really relevant for the fundamental research, the basic research, but also for the drug development. Organoids, as I mentioned, can be grown from the patient's cells directly. So basically can be used for precision medicine or for to better understand the mechanism of a disease with a mutation of the genetic mutation that we're finding in patient. And there's a lot of very other relevant application for drug discovery, etc. And I wanted to give you the example of how successful the organoids technology was, especially for the study of viruses. And there is a nice example of a study that used lung and brain organoids to better understand the mechanism of the Zika viruses. And thanks to this study, they understand better how they what was the mechanism under the microcephaly that were observed after the Zika infection. And more studies are using organoids, for example, to better understand the impact of the SARS-CoV or the COVID on the different organs of the human body. A very interesting project that is ongoing and that shows how powerful the organoid technology can be is the Organoid Cell Atlas. So the project aims to put together a living biobank with all the information of all the patients and all the data from the organoid research, confronting the data from the patient and the data from the organoid to validate the tool and also to improve it. And the ultimate aim of this project is to apply the organoids, being able to apply the organoid technology to the disease understanding process and then to the treatment, the development of new drug, and to the assessment of the mechanism behind the drug. And finally, in the in vitro field, there's like a revolution that is linked to what we call the organ on a chip, which is also called the microphysiological systems, that are basically combining the high quality of engineering with the biology, and basically these systems are microchips that contain channels and different channels and different for example, what we call barrier. So basically it allows to recapitulate something that you don't have, for example, with organoids, is the flow, so the movement. You can recapitulate interaction between different organs, like the blood and the lung, or you can also recapitulate the blood, what we call the blood-brain barrier. So interaction between the blood and the brain, which is really important, and it has been limiting so far because it was really hard to study. So the organoid chip is a really versatile tool because basically you can grow whatever you want inside this microchip. You can use a cell line, you can use what I mentioned earlier, the induced pluripotent cells, but you can also use micro tissue, organoids, and 3D printed tissue that I will mention later. And you can basically grow these cells or in different environments with a scaffold or without a scaffold. You can put some barriers to mimic the barrier in vivo, and you can put a fluid flow. Basically capitulating the, for example, the blood or the airway. So this is like really like revolution for the in vitro field. It's bringing much more complexity, which is really important to mimic human body. And what is really powerful with the organ and chip as well is the fact that you can combine the microchip. So basically, this is hugely relevant if you want to understand interaction between organs. And you can even combine multi-chips to recapitulate basically all the organs of the body. That is called the body on a chip. There's a couple of examples of really like striking and successful studies using the organoid chips. For example, this study from the Emulate company is developing chips, and an hospital in the US, and they use a patient, like what they call the patient on a chip for the gut, studying the gut diseases. And they were able to recapitulate really the polarization of the cells exactly like it will happen in the body. And it recapitulates as well the different cell type of the intestine and the functionality of the intestine. So, for example, they were able to observe the responsiveness of certain stimuli, which is really striking and promising for future research in gut diseases. And another very, very relevant study that has been published really lately with the emulate liver chip DILI research, so for the drug-induced liver injury. As you probably know, one of the main reasons why a drug is failing in clinical trial is the toxicity to the liver, and the fact that it wasn't predicted in animals before. What the study has done is to test 27 known drugs that are toxic or not for the liver in a lot of different chips. And they've been able to show that actually the liver chips has a sensitivity of 87, which is like much higher than animals, and the specificity of 100%, which obviously is a great score. And compared to animals, which is 0, it's really a striking and revolutionary. And they calculated the costs, the benefits of using liver chips for the drug development, and they were able to estimate that using this technology will generate 3 billion annually for the pharmaceutical companies. And obviously, there's if you look at the drug development pipeline from the discovery to the market, all the tools that I have mentioned can be used, basically because they all have advantage or limits. And depending on the stage of the study and the development, obviously in the beginning you want to screen the drug, you want to find nice targets and a nice promising drug. So you need to go to a high level of testing, what you call the high throughput. And for that, the 2D cells is like induced pluripotent stem cell in 2D are very relevant because they're cheap and they are really efficient and reproducible. And then you can move to models that are closer to the physiological relevance in the human body, like the organoids, and then finally the organ on chips, multi-organ on chips, and body on a chip. So hopefully with like a more standardization of all these protocols and all these tools, we will be able to show that actually we don't need anymore the animal to predict the drug efficacy and to then test them on human. I've already mentioned that, so maybe I will go through quickly to this to this slide, sorry. But what I wanted to really insist on is like, there is not a good model. It's really depending on what you want to study and what you want to look at. So all in vitro models are really interesting, relevant, and it's really depending on what the study wants to show, what is the question behind, and how you want to study it. So as I mentioned, the 2D, the 3D, and the 2D static models, without any fluids, are more interesting for the throughput, so for the high throughput screening of a drug. And then if you want to better understand a disease or mimic the disease, it is much more relevant to use the organ on chips or even the multi-organ on chips, etc. Something that is to mention is that even though the organ on chip technology is revolutionary, it still has some limitations. So as mentioned, it's quite hard because it's really technical to use. It's not yet suitable to the high throughput screening. Hopefully it will be in the future. And what is really I think holding back the technology is the fact that it doesn't have any valid protocols. And you probably heard earlier that this was an issue with animals. So if we want the in vitro model to better predict human diseases and be better than animals, it is really important to develop and validate protocols that are reliable and that will lead to the fact that these models are less fireable and more reproducible, so more relevant as well. And the cost of these technologies is still high. So obviously it's getting cheaper and cheaper over time, but it is really important to take that into account and to consider the fact that it is an investment. And so if you want to invest in that, you need to know that it's expensive, but obviously less expensive than using animals. I also wanted to quickly mention the revolution of 3D bioprinting and tissue engineering for the study of diseases. It's really something that's getting more and more used and getting attention also for the research and development of drugs for diseases. Because you can use 3D bioprinting to grow organoids as well. You can use the technology to produce some biomaterials like hydrogel and the scaffolds as the important basis for developing growing the organoids. 3D bioprinting can also be used to recreate organs and real-size organs that can be in the future useful for regenerative medicine, for organs transplants, and also for drug testing. 3D bioprinting is also used to print the chips for the organochip system. It can also bring the organoid itself, as I mentioned. And you can use patient cells to print new organoids or organ, which is obviously more relevant. And some studies have shown that in cancer, for example, you can basically 3D print your tumor with the microenvironments. And this is really relevant for example, for high throughput drug screening. Just like the organ on chip technology, the issue with bioprinting is the fact that it hasn't been really validated yet. We don't have any standard for that, and it needs to be optimized because there is an issue also with the viability of the cells. As it's like it might be stressful for the cells. So it is still under development, but it has really great promise and should be considered as well. So I've mentioned all not all, but hopefully I've mentioned the main in vitro new approach method that holds great promise. But I wanted to show you attention on the what we call in silico, so computer technologies, big data, artificial intelligence, and computer modeling that come now as a real promising tool understanding of diseases and development. So it has been more and more obvious that using what we call the omics approach, which is basically taking a sample of a cell or a tissue for humans and sequencing this information from the cells. It can be the genetics. It can be what we call transcriptomics, so the genes themselves, the proteomics for the proteins, or metabolomics to study the metabolism of the human body. All these technologies are now well established and used and hold great promise. They can provide a significant amount and very interesting information to study and better understand human diseases. It can be applied to find biomarkers, what we call biomarkers. So, for example, in cancer, it will be some key genes that are driver like that will lead to the development of the cancer. it can be also the gene that are identified as a poor predictor for disease. For example, if there is a mutation in a gene, the patient will be more likely to relapse, or on the opposite, to respond to a drug. So it's really interesting and essential to use now the omics information as a complement to the in vitro approaches, and it holds a great promise of to better predict a disease, to better diagnose a disease as well, and to identify the molecular mechanism of a disease as well. And also all these big data that are generated by the biological field using patient data are now applied and used more in depth with what we call the artificial intelligence and computer modeling. That has a lot of key application. It can be used, for example, to better analyze some data, for example, some imaging data that, for example, an eye of a human couldn't see, and also to analyze a big amount of data that would take ages for a human, and to combine all this information to use what we call the deep learning. That will just basically be able to predict, like, for example, a toxicity of a drug or an efficacy of a drug. For example, also it can be if the data that you provide to the computer is like the structural information of a drug, it will be able to predict the pharmaceutical property of these drugs. So how it will be incorporated into the body, will it be toxic or not to the body, and so on and so forth. And as I mentioned, also it can be used to, for example, I can give you an example of how artificial intelligence is really useful for disease diagnosis. So it has been shown that artificial intelligence is really reliable to predict and diagnose the melanoma. So if you take a picture of a human skin with the possibility of a melanoma, the artificial intelligence will be more reliable to say if this is like malignant or not, and to predict the evolution of the disease as well. Computer modeling is also used more and more in the pipeline to optimize the drug development and the patient treatment. It can help anticipating the efficacy of the treatment depending on the patient and depending on the genetic background of the patient as well. So I hope that it was a comprehensive library of what we call the new approach methodologies. This is like an overview of what I've already mentioned. And I wanted to emphasize the fact that what is really powerful, I think, with the new approach methodologies is the fact that you can combine them. So they are really complementary. And as mentioned, there is not one good model, but what is really powerful is to combine the model to make sure that it is reliable and promising and reproducible as well. So I already mentioned briefly that during the talk, but I think that the thing that is the most important with new approach methodologies is the fact that they don't seek to provide a replacement like for like from animals. As I mentioned, animals are not really good. So it's not here to replace it, but it's here to provide better information that are more relevant to human and that has a greater chance to be relevant for the understanding of mechanism of diseases and of drug efficacy and toxicity. And the biggest challenge with the new approach methodologies, and I think I already mentioned that quite a lot, is the need for validation and standardization of this technology to make sure that they are reproducible, that they are well characterized. And something that is really important as well is the fact that it's coming together with the validation and standardization, but it is really important that scientists that are willing to use this technology are well trained. And also, if there's more and more scientists using the new approach methodologies, they will bring more data to showcase their relevance. It will help bringing more awareness as well. And it will likely and hopefully change the bias that a lot of scientists have regarding animal experiment, the fact that they always think that it is the best approach to study the diseases. Hopefully that will change too. And I wanted just to finish with a positive note, because I think it's really important to also acknowledge the fact that I think we're getting closer to the acceptance of the new approach methodologies. And it's well illustrated by what has been recently passed at the US Senate, what we call the FDA Modernization Act. Basically, this bill allow a drug to be approved to go to the market, even though it hasn't been tested for animal, meaning they are accepting the fact that some alternative methods are as good or even better than animal, like what I mentioned earlier, the organ-on-chip or the computer modeling approaches. And even though it's not like telling it's not like pushing science to completely stop animal experiment, it's allowing the use of the alternatives, and I think it's a great progress that we should be happy and exciting about. And also something that was announced really lately, and I found that really exciting too, is the fact that a pharmaceutical company, Roche, has launched an institute, what they call the Institute of Human Biology. So basically, the institute will focus only on human-based methods. They want to bring together scientists from academia and industry, and the idea is to lead to a broad adoption of the human model system by the pharmaceutical research and development field, and also to accelerate the breakthrough of the drug development and use this human model system to better predict drugs and efficacy and develop better human model system. So it's really positive, and I wanted to just mention that. I hope you could hear me well, because I just had the feeling that my internet is not great. So hopefully you could hear me well, and this is all for me. I just wanted to thank you very much for being here today. I wanted to mention two important things that we are at Animal Free Research UK happy to shout about. Is the science conference that is going to happen this year, end of the year in October in Cambridge, and the Community of Practice platform, that is on an online forum for scientists to discuss, interact, collaborate, all about the human-based methods. So thank you so much for your attention, and I'm happy to take any question you may have now. And I think think I was a bit too long. Sorry about that too. Oh, I can see a question from Sam. Which technology are you most excited about? I think that will be, I mentioned the combination of technology. I think the organ on chip is definitely the one I'm currently most excited about. I think it's only the beginning of this technology and its development, and it holds so much promise. So yeah, I would say the organ on chip. Oh, thank you, Lisa, for these comments. This is actually really important. I didn't have time to mention, but hopefully next time I will mention it. So she's asking about the animal-based materials. Obviously, all the technology I presented are still using probably for most of them using animal-derived biomaterials like hydrogel, serum and stuff like that in antibodies. So this is also something that should be considered, I think, really during the standardization protocols and validation of the tools. Hopefully, and that's something that we are also really willing to push. We are keen on developing tools for researchers to be able to find the best animal-free materials for the in vitro approach. I'm sorry again for being too long. I hope that it was still okay. And if there is no more question, I think we can wrap up.